My name is Ajda Gurkan from Marmara University, Istanbul, Turkey. First of all, I would like to thank the organizing committee to give me a chance to present my research. My topic is synthesizing the Aptamers against growth hormone releasing hormone and its characterization in vitro analysis. I hope that I can pass the slide by this way. As all we know that cancer is a complex disease and various steps have been performed during the different signaling and the alterations of protein and transcriptional levels. And it will be a deep problem in all around the world for various cancers to prevent and also detect. And various novel approaches are trying to cure the disease, but somehow it's a little bit very hard as it's a more complex disease. And I try to work on especially hormone dependent cancer progression and also the cancer drug resistance due to the hormone expressions. As one of my target is growth hormone releasing hormone. It's a hypothalamic neuropeptide, generally targets the pituitary gland for the expression of the postnatal growth hormone. But when you look at the literature, there are lots of expression studies of growth hormone releasing hormone and its receptor in various tissues, peripheral tissues, such as prostate, breast, ovarian, and endometrial. And also in the biopsy samples of these cancer types, the expression of the immunohistochemical analysis also performed. As we know that you can see on the left side the peptide sequence of the growth hormone releasing hormone, and we know that in the 20th century that generally neuropeptides, especially the analogs, are designed and characterized for the anti-apoptotic and anti-cancerogenic effect in vitro, in vivo of growth hormone releasing hormone, especially in the prostate, colon, and also breast cancer. From this point of view, we try to figure out whether we can perform aptamers against growth hormone releasing hormone. And we know that aptamers are specific target nucleic acids. They have a high affinity and they are non-immunogenic, and also they have a serum stability in vivo. So we know that macugen is the one of the most popular aptamer against macular degeneration. And we try to figure out whether we can perform aptamers against growth hormone releasing hormone. And instead of this point of view, we try to figure out its anti-cancerogenic effect in colon and pancreatic cancer cell lines in vitro. During this point of view, we have, of course, the first target. We synthesize, both two plasmids that's expressing the growth hormone releasing hormone with the his-tagged protein. After purification of the protein, as you see on the figure 1A, and we perform that the bacteria have a large amount of this protein. So we generally also perform the stable cell line in HEC293 cells for the expression of the growth hormone releasing hormone in the RT-PCR on the C side, and immunoblotting in the B side, and immunofluorescence assay on the E side. And we use this target. For this point of view, we perform an Aptamer kit. According to the kit, they are beads, and the Select pool, DNA pool is on bounded. And we perform cycles of PCR, and we send the PCR products to the company, and they synthesize us 3-4 different aptamers after NGS sequencing. And on the right side, in the C part, you can see the sequences of the aptamers against growth hormone releasing hormone. And we try to perform their secondary structure as the aptamers have some putative different base analogs. So it's a little bit hard for us to perform their KD and delta G levels. And we try to start to perform their especially kinetic energy performations. And first of all, we determine the dot plot analysis, as all we know that, to determine the binding efficiency of all these aptamers. And we perform, some of them have a high binding affinity against the target. And we try to perform them in dose dependent manner in the dot plot analysis. And some of them have a specifically high binding affinity. Later on, in order to perform them in the in vivo studies, we perform the human serum incubation periods in a time-dependent manner, and the targeted, especially the important aptamers, have a high binding affinity and also high serum stability. And later on, we perform the CPR analysis of our tree target, as we know that is expensive experiment for us. And we get 3 of them have a low KD levels. Later on, we try to figure out the in vitro studies. First, we have to determine the growth hormone signaling, releasing hormone signaling, and we use MIAPACA, HT29, LNCAP, PC, and PNT1 cells. In the immunohistochemical analysis, immunofluorescence assay, you can see the green dots as an expression of these hormone. On the B side, you can see the various 2 different variant expression profile of this, and the ELISA assay give us that all these cancer cell lines types have an expression of growth hormone releasing hormone. Then we try to perform the co-immuno binding assay in the fluorescent microscopy against the growth hormone and growth hormone releasing hormone, as they are the important expression profiles. And the after binding the aptamers on the growth hormone ligand in order to block the growth hormone releasing hormones signaling, we perform that some of the aptamers have a potential inhibitive activity on this signaling pathway. As we know that cyclic AMP is the major target secondary molecule in the G protein coupled receptor. One of them is the growth hormone releasing hormone receptor, and we perform that in the MIAPACA and H229 cells have degrees secretion and catalytic activity of cyclic AMP. And later on, we try to figure out the binding affinities of these surface receptors and the ligands, and we understand that all these aptamers have a affinity against the target, and but they have an important epitope to bind to the receptor and blockage the signaling pathway in various cancer cells, especially the colon cancer and also prostate cancer. You can see the light microscopy image and also the against the streptoiodine alexaflor binding affinities of these aptamers against. So later on, after these binding characterization activities, we perform the MTT assay and to determine the colon cancer and also the prostate pancreatic cancer cell lines effect of the aptamers against nuclear condensation and mitochondrial membrane potential loss. And we determine that these especially 4 aptamers have a specific effect on these cell lines, as you can see on the MTT on the right figure, and also fluorescent microscopy on the left side. And also we try to figure out the effect of them on the flux flow analysis, and we try to figure out whether they have an effect of early or late apoptotic induction. And we use as a PNT1 cells as a normal epithelial cell, and we perform that significant G2M cycle arrest of these aptamers in the cell cycle progression of these cancer cells, and we try to figure out whether they have an important impact in vivo, and we have give a project about it. So we detected and also amplified X aptamers, single-strand DNA aptamers, and we have 24 putative ones, and 5 of them have a high binding affinity against the growth hormone releasing hormone peptide, and 2 of them especially give a significant inhibitory effect on cell viability in HT29, colon cancer and Mayapaka pancreatic cancer cell lines. And they have a significant effect on the cell death via Fox flow analysis. We published these results in the PLOS One and can get more results there. And I would like to thank my colleagues and also my graduate and undergraduate students for their help and technical helps, and I would like to thank to all for your attendance to my research. Thank you.